Hi everyone, today I'm going to solve this paper from Mock Championship 2026 by Fast Papers, and this is Cambridge O-Level Biology paper 2 and the total mark is 80. Question 1. Figure 1.1 shows two cells from the leaf of a plant observed using a light microscope. 1a. Table 1.1. Complete table 1.1 to identify the structures labeled A and B and to describe the function of each structure. So structure A is clearly the sap vacuole, or you can just write vacuole, and it acts as a storage and provides starter and support to keep the cell firm. And B is chloroplast, which are these ones over here, and these are organelles that contain chlorophyll, so it helps the plants in photosynthesis. And you can also write it converts light energy into chemical energy to produce glucose and starch for the plant. In B, state one structure that is not visible in figure 1.1 but would be visible in the cytoplasm in an electron microgram of the same two cells. So I'm going to provide a diagram over here. You can see clearly what's missing. So either of two are the answer, which are ribosomes or mitochondria. So you mention either one to get 1 mark here. Moving to question 2a. Hormones are produced by glands in the body. They alter the activity of specific target organs. So draw lines to link each gland to a hormone it produces and each hormone to a specific target organ. So firstly we have adrenal, and it produces adrenaline, which is produced in stressful situations. It causes your pupils to dilate, your heart rate increases, and your breathing is faster. So the target organ is the heart. Next we have ovary, and it produces progesterone, and the target organ is obviously uterus because its function is to create a uterus lining and keep it thick. And when menstruation starts, this hormone level drops. Next we have pancreas, which produces glucagon, and the target organ is the liver. What glucagon does is it converts glycogen into glucose when our blood sugar levels are low, and it's important to not confuse this with insulin, which converts glucose into glycogen. And the target organ for this is liver because this is where assimilation of digested food occur. So this is where glucose is produced. Lastly, the pituitary gland produces FSH and also LH, and the target organ is ovary. So what it does is it stimulates the growth of eggs and also forming corpus luteum. So when this happens, the ovary then triggers the production of progesterone. In 2b, name a chemical that is produced in a plant shoot to coordinate the shoot's response to gravity. So the answer is obviously auxin, because this is found in the tips of actively growing roots and shoots, and it responds to light and gravity. Question number 3 is figure 3.1 shows a section through the human eye, and I'm going to include a diagram on top. So 3a1, identify the letter on figure 3.1 that has been used to label the part of the eye that contains circular and radial muscles. And as we can see in the diagram, this will be the letter K because it's iris, and iris also contains circular and radial muscles. Next has the highest concentration of light receptors, which is N, and that can be seen clearly in the diagram. This is because the structure N is responsible for detecting a lot of colored light. Okay, so next on figure 3.1, draw and label the lens and the suspensory ligaments. So I drew it here. This part here is the lens, and this two over here are the suspensory ligaments. And I'll show you the diagram one more time so that you can see the accuracy of it. Okay, then in A3, explain the function of suspensory ligaments. So again, if you look at the mark scheme, the answer has more than 3 mark worth of points. So I'm gonna explain all of them. Suspensory ligaments are attached to the ciliary bodies in the eyes and can either be loose or tight. So these are the ciliary bodies, and for example, when the ciliary muscles relax, the suspensory ligaments tighten and the lens is pulled thin. This is done so that the eye can look at something that is distant. So this is also known as accommodation for distant objects. So the suspensory ligaments pull on the lens to control its shape. They also allow accommodation to occur, meaning it can change its focus on different objects at different distances. So in contrast, if you are looking at something that is near and not distant, the ciliary muscles will contract and the suspensory ligaments will relax and the lens will also relax. So this is an accommodation for nearer objects. In B it says cornea plana is a genetic disorder that results in an abnormally flat cornea in affected individuals. So B1, explain why a person with cornea plana cannot see some objects clearly. So what a cornea does is it refracts light and creates an image. So in this disorder, the light will not be refracted enough and not be focused on the retina, making objects unclear. So the image that's being formed as clear as it normally would. One type of cornea plana is caused by a recessive allele. Two people who do not have cornea plana have a child who has the condition. Explain how this is possible. So we move on to genetics a little bit. Both of the parents must be heterozygous, meaning both carry one affected allele. Therefore, there is 1 in 4 or 25% chance of the recessive allele to be passed on, which is what led to cornea plana in this case. So you can quickly form, let's say, the dominant allele is a C and recessive allele is a small c, and the parents have these as their heterozygous. So let's say you would take the genotypes as a capital letter C and a small letter c, and you draw a quick diagram of you draw a quick Punnett square and you put the genotypes as such. Sorry, this is small. So there's 1 in 4 chance that this can be passed on, indicated by the small letter C's, and whenever you have the bigger C's, it's not possible for the person to have cornea plana. Only when you have the 2 small letter C's, you have 25% or 1 in 4 chance for this to be passed on to your offspring. Moving on to number 4, this is pretty simple. Organisms can be classified into groups using features they share. So 4A, fish are one of the main groups of vertebrates. 1. State 3 main features used to classify an organism as a fish. So we have gills, scales or scaly skin, fins, but you can also mention either of these points. So they have eggs with no shell or surrounded by jelly, they're cold-blooded, and the fertilization happens externally. 2. State 2 main groups of vertebrates other than fish. So either of the 4 can be mentioned to get 2 marks. Either 2 from the 4 can be mentioned to get 2 marks. So either mammals and birds, or reptiles and amphibians, or any mix of the 4. B. Swordfish are large fish that live in the ocean. They have a long bony extension to the skull called a sword. Figure 4.1 is a diagram of a swordfish. The binomial name of the swordfish is Cepheus gladius. I am not sure how to pronounce it, but let's go on with it. Define the term species. So it's a group of organisms that reproduce fertile offspring. So it's simple as that. So I think 1 mark goes to the group of organisms and reproduce and fertile offspring. So state the species name of the organism shown in figure 4.1. So we get gladius from it. Okay, so Swordfish swim near the surface of the ocean where water temperature is low. During evolution, swordfish developed a muscle behind each eye that warms the eyes and the brain to a temperature above that of the ocean. This feature gives swordfish improved vision. So, C1, name the process that led to the evolution of the muscle behind each eye of the swordfish. So, obviously, since it's an evolutionary feature, it's done by natural selection, which happens when the more adaptive features are when fish with the more adaptive features mate and produce an offspring which gets passed on, and they evolve in that way, so they have better vision and other qualities. Suggest reasons why the evolution of this feature has adopted swordfish to survive in the ocean. So now we're going to elaborate a little more on that. This allows better sight to hunt prey for food. Again, please look at the marking breakdown. Food allows more energy and gives a competitive advantage against other animals. This feature also aids them to see predators in order to avoid being attacked. So they're being better as a predator, and they're avoiding other predators that would hunt for them, and this makes their survival much more efficient. Moreover, search for mates can be efficient for reproduction and increasing population. So like I previously mentioned, a better quality, a more evolved quality is of a certain animal can make it better for them in finding mates, so that these good qualities can be passed on. Moving on, number 5, the castor oil plant is a flowering plant which produces seeds. The seeds have an additional structure called an oil body that contains oil. So it's this part over here. And figure 5.1 shows a castor oil seed with its oil body. So in 5a, describe a laboratory test that can be used to show that the oil body contains oil. So naturally we're going to go with the emulsion test, and we're going to start by adding ethanol to a seed in a test tube or any other container, and then by adding water. And if the mixture turns cloudy or white emulsion has formed, the test is considered positive for the presence of oil or fat. Look at 5b. When the fruits of the castor oil plant are ripe, they burst open, scattering the seeds onto the soil. Ants feed on the oil bodies, but not the rest of the seeds. They either remove the oil bodies where the seeds have fallen, or they take the seeds and oil bodies back to their nest. Colonies of 5 different ant species, A to E, were supplied with the same number of castor oil seeds near to their nests. Figure 5.2 shows the results of this experiment into the behavior of these different ant species. So the darker area of the bars indicate that oil body and seeds taken to the nest, and the white part shows that oil body is only taken to the nest. So just to break it down a little bit, the ant species A has mostly taken the oil body and the seeds to their nest, whereas B, only some of them just took the oil body, but they mostly didn't take anything back to their nest. C, on the other hand, not all of them had taken the seeds and oil bodies to the nest, but most of them has taken both, and a little percentage of them has only taken the oil body. So the other two is pretty self-explanatory. D and E, only a small proportion of them have taken both. All of E has taken either, but they mostly took only the oil body and not the rest of the seeds. Only a small number of flowering plant species produce seeds with oil bodies. Suggests one advantage and one disadvantage to the castor oil plant of producing seeds with oil bodies. So the advantage is it increases seed dispersal as the seeds are carried by ants, and the disadvantage is it requires more photosynthesis in order to make energy for lipid synthesis. So the plant needs to respire more and for longer for the photosynthesis to occur and make energy for the oil production to occur. Okay, so the next part is use information from figure 5.2 to explain how useful the different ant species are to the castor oil plant. So, like we discussed, species A and C are the most useful as they are carrying the seed along with the oil body. But species B, D, and E are not useful for dispersal as seeds need to be removed and not just the oil body. 3. The ants feed on the oil bodies and digest the oil. Name the products of this digestion. So, we take oil as fat, and fat is broken down into fatty acids and glycerol. So this is all for this answer. And then in C, ant species A is Solenopsis geminata. Figure 5.3 shows a diagram of S. geminata. So this is an arthropod. The arthropods are classified into a number of groups. State the name of the group to which S. geminata belongs. So it's obviously an insect. State 2 visible features used to classify S. geminata in this group of arthropods. So number 1 is 3 pairs of legs and legs attached to thorax. So there are also other acceptable features. So you can either mention 3 sections to body, or they have a head, thorax, and abdomen, and 1 pair of antennae. So there are 2 of them here. So these are the features that you need to identify this group of arthropods. Alright, question number 6. A student investigates the use of an enzyme in fruit juice production. The student carries out the following steps. So the student cuts 1 apple into small pieces, puts the same number of pieces into each of 3 beakers labeled A, B, and C. Then adds 2 centimeter cube of a dilute enzyme solution to the apple beaker in beakers A and B, and adds 2 centimeter cube of distilled water to the apple in beaker C. Then places each beaker in a water bath at a constant temperature for 30 minutes. Filters the juice from each beaker into separate measuring cylinder and records the volume of juice collected in each measuring cylinder. So identify the enzyme that the student should add to the apple in beakers A and B. So the answer is pectinase. So this is the answer because pectinase is a group of enzymes that break down pectin, which is a polysaccharide that is found in plant cell wall. Outline the reason why the student set up beaker C. So beaker C had 2 centimeter cube of distilled water, and naturally beaker C was placed as a control because you want to see the effect of enzyme and to make sure that whatever you get after the water bath is because of the enzyme and not because of anything else. So whatever you get in beaker C will be the result of no enzyme being present. So there has to be a difference between beaker C and the other 2 beakers A and B. In B, bacteria can be genetically modified to produce enzymes for use in biotechnology. State 2 reasons why bacteria are suitable for this purpose. So there are a number of points here. You can mention either 2 to get 2 marks. So bacteria contain plasmids. Bacteria are able to make complex molecules, and because of this, they produce genetically identical products. So it's helpful as it is more identical to other living beings, humans, and it's a good measure for biotech because causing harm to bacteria is not considered as an ethical concern. And also bacteria are able to make complex molecules at the same time, and they usually reproduce very rapidly, and they are easy to grow as well, which makes a win-win situation for biotech to use bacteria for producing enzymes. Okay, so in C, we see the results of the student's investigation, and this is what we get after the water bath. So the temperature in A was 45 degrees, in B it was 90 degrees, in C, which contained distilled water, so C is the control, and the temperature was also 45 degrees. So in beaker A, we see that 20 centimeter cube of juice was collected, and in the other 2 there was 5 on each. So use your knowledge of enzyme action to explain these results. So beaker A produced more juice in comparison to beaker B, obviously. There's a huge difference, and also beaker C. So in beaker A, enzymes are working at 45 degrees Celsius because it has produced 20 centimeter cube of juice by having effective collision to the active site. And in beaker B, less juice have been produced. Basically, since this is a control and it has produced 5, this is also 5, so it shows that enzyme has barely or didn't even work. So this is due to high temperature of 90 degrees Celsius, and the enzyme was denatured and the substrate no longer was able to bind to the active site. So this is why the control was important, to show the effect of temperature and the enzyme. The enzyme's optimum temperature was probably 45 and 90 was too high, so barely anything was produced. And in control, there was no enzyme activity, so this was also fine. Moving to number 7, the nucleus of a cell contains DNA molecules that control cell function. Figure 7.1 is a diagram showing one of these DNA molecules. So on spot we can see that Q is a chromosome, so In A, using information from figure 7.1, state the name of the unit of DNA labeled P. So this will be a nucleotide. It's pretty simple, easy to identify. And in 2, state the name of the structure labeled Q, which is chromosome. And in 3, state the letter of the base that pairs with G. So we know C pairs with G and A pairs with T. B, explain how this DNA molecule controls cell function. Include an example in your answer. So what DNA does is it codes for polypeptides or proteins. The mark scheme mentions both, and it's better to mention polypeptides because this is what makes proteins, and DNA does not directly make protein, is what I'm saying. So the base sequence determines the sequence of amino acids. So polypeptides are a chain of amino acids that act as building blocks of protein. So the amino acid sequence is where the DNA for specific proteins are determined. So amino acids basically make up different proteins. So what happens here is the DNA codes for polypeptides, the amino acid sequence arrange for those polypeptides, and then proteins are formed. Okay. Number 8, the carbon cycle plays a vital role in all ecosystems. Carbon is continuously transferred between organisms and the atmosphere, as shown in figure 8.1. So carbon is going from atmosphere to living organisms and then from living organisms back to atmosphere. Describe how carbon is cycled between living organisms and the atmosphere. So carbon dioxide in the atmosphere is absorbed by producers such as plants for photosynthesis. Carbon dioxide is taken in by the chloroplasts. Plants use it to produce carbohydrates and organic substances such as fruits. These organic matter is eaten by other living organisms, basically consumed by animals, and therefore transferred to them. As animals respire, they release carbon dioxide and it goes back to the atmosphere. It's a very simple answer, but again, refer back to the mark scheme to see what points are enough for you to get 5 marks. But aim to answer a little bit more so that you can be sure about your answer. In 8b, woodland ecosystems are frequently managed by humans. When trees are cut down in a woodland, they may be burned, taken away, and used to make products, or left within the ecosystem. So compare the effects of these 3 different forms of management on the woodland ecosystem and more widely on the planet. So first, let's address this one. Burning releases carbon dioxide, causing a greenhouse effect, which adds to climate change such as high temperatures, global warming, and so on. Products that are made from trees act as a carbon store. Since the trees are removed from the ecosystem, so now we're addressing this, it also removes the nutrients that are stored in the plants. Trees. When cut trees are left within the ecosystem, they are fed on by decomposers such as fungi and bacteria, and therefore nutrients are returned to the soil. So now they're being taken away from the trees and being returned to the soil by the decomposers. This also means carbon dioxide will be gradually released as decomposers respire. So this makes dead trees a habitat for this organism. So basically they're getting what they want from the dead trees, so they start to create their own habitat in that area. Okay, so now at the last question. Pectinase is an enzyme that is produced by fungi. In the large-scale industrial production of pectinase, fungi are grown in fermenters. So I'll show you a picture of what an actual fermenter looks like. So it's better to always look at the labels of the diagram before you start answering. So it will save you some time, allow you to think, and you don't have to come back to the diagram again and again. But you should be familiar with the diagram already of how a fermenter works. Explain how the features of the fermenter shown in figure 9.1 provide suitable conditions for the growth of fungi. So another answer that includes all the points. So what does a mortar do? It mixes this solution, ensuring food supply, heat and air, and waste products are distributed evenly so that there is no sedimentation of waste or food, and heat is evenly spread through the solution so that fungi get access to everything at once. So the task of the water jacket is to cool the fermenter. So it brings water around the fermenter and helps to cool its contents. As a result, high temperatures cannot kill fungus or denature enzymes. The heated water then moves out. So we have water out here. So water comes in, cools the contents, and then water goes out. And the motor obviously mixes everything. A supply of air into the fermenter provides oxygen for fungus to respire aerobically. This air has to pass through an air filter, which prevents the entry of microorganisms. So you can just let the normal air in. It has to pass through a filter so that nothing else that would interfere with the fermentation, such as microorganisms, are coming in with the air. This prevents the solution in the fermenter from being contaminated, as well as reduces competition for the fungi. So this will be our last question of the paper. In an experiment, scientists added pectinase made by the fungi to a sample of crushed fruit. They measured changes in the sugar content of the sample of crushed fruit and the thickness of the sample of crushed fruit. So in this graph, we can see the thickness, which is indicated by this line, and the sugar content, which is indicated by this line. So from a pattern, we can notice that the thickness reduces when the sugar content goes up. And this is where pectinase is added. So the whole of the graph shows what happens after it's added. So use your knowledge of the action of pectinase to explain the scientist's results. So this is another detailed answer. Pectinase breaks down and digests pectin, releasing sugar from pectin. This increases the sugar concentration. As pectin makes the solution thicker, the thickness decreases when pectin is digested. So again, look at the trend here. When sugar content is increased, so when more pectin is digested, the thickness decreases as indicated by this line. This allows sugars to reduce the thickness when they are dissolved. So the thickness is further reduced when the sugar gets dissolved as well. Okay, thank you for watching. Best of luck for your exam.